is a commonly used analytical biochemistry assay. The assay uses a solid phase enzyme immunoassay to detect the presence of a ligand, commonly a protein, in a liquid sample using antibodies directed against the proteins to be measured. ELISA has been using as a diagnostic tool in medicine, plant pathology, and biotechnology, as well as quality control check in various industries. ELISA is also commonly known as EIA or just the enzyme immunoassay. So now let's break the term ELISA and understand its meaning. So the EL of ELISA denotes the enzyme is linked with the detection antibody. IS denotes an antibody or antigen is adsorbed onto the microvelles in which we conduct the tests. And A denotes quantity of a specific antigen or antibody in the sample. So basically what happens is the antigen or the antibody which we are detecting in the sample bounds with the enzyme-linked antibody or the antigen. This complex, when reacts with a substrate, gives color. Say for example in direct ELISA, the antigen from the sample bounds with the enzyme-linked antibodies. Then when we add a substrate, this substrate then reacts with the enzyme and gives a characteristic color. So this is how ELISA works. We will discuss each type of ELISA in detail later in the lecture. So the principle of ELISA is ELISA uses the basic immunology concept of an antigen binding to its specific antibody, which allows detection of very small quantities of antigens such as proteins, peptides, hormones, or antibody in a fluid sample. So ELISA assays are usually chromogenic, using a reaction that converts the substrate, for example tetramethylbenzidine, into a colored product which can be measured using a plate reader. The most commonly used enzyme labels in ELISA are horseradish peroxidase, glucose oxidase, and alkaline phosphatase. So these are the enzymes which are linked with the detection antibodies, and the substrate we use in the technique of ELISA is TMB or tetramethylbenzidine. So in this picture you can see that there is an antigen-antibody complex. The antigen is bound by a capture antibody and an enzyme-linked antibody. And when these both two react, when the antigen and the antibodies makes a complex, on adding the substrate, it gives us a color. So let's just get an idea. I hope you all must have been cleared about the antigen and antibodies, that what antigen and what antibodies are. Antigen basically is a substance that when introduced into the body, it stimulates the production of an antibody. For example, any bacteria, viruses, or fungi, these are all pathogens. Okay, whenever such proteins or such pathogens enter in our bloodstream, our body's immune response makes antibodies against those antigens. So antibodies, also known as immunoglobulins, are found in blood and are produced in response to antigens. So this is quite simple. Antigens and antibodies are both they both make complexes, and we detect these complexes and these things separately while doing the ELISA and other different techniques for the detection of for the diagnosis of multiple diseases. So this is an ELISA kit. You can see here this is the 96 well plate, and this is the reservoir and a 12 channel pipette, and this is the plate reader. So these microvelles, these are the microvelles which are coated either with an antigen or an antibody, depending upon what type of test we are performing. So the types of ELISA. So there are basically 4 types of ELISA: direct, indirect, sandwich ELISA, and competitive ELISA. So before discussing the topics separately, and let me just first clear the concepts of primary and secondary antibody here, so it would be easier for you to understand these types of ELISA. So the primary antibody is that antibody in which is antigen specific, meaning it is produced in response to a particular antigen. For example, a patient is suffering from hepatitis A. Okay, so hepatitis A is the antigen, and the patient's immune response produces antibodies against that specific virus, which is the hepatitis A virus, and those antibodies are specific for that particular hepatitis A antigen. So those will be called as primary antibodies. I hope I'm clear now. So these primary antibodies are taken from human serums. Okay. Secondary antibodies are synthesized in the labs by the introduction of the primary antibody to an animal species. So now secondary antibody basically is synthesized by the human beings. It is synthesized in the laboratory settings. What happens when we capture those primary antibodies from human serum, and we inject those primary antibodies into the animal host. So for that animal host, those primary antibodies are acting as antigens, because for those animal species, that antibody basically is a pathogen or any foreign object or the antigen. So for that particular antigen, the animal's immune response or the immune system makes antibodies against those antigen, which are originally primary antibodies, right? So these newly formed antibodies from the animal species are then collected from the serums and are used in the process of ELISA. So this is the main difference between the primary antibodies and the secondary antibodies. So now coming back towards the types of ELISA, which are direct, indirect, sandwich, and competitive. So the direct, these definitions are quite short. We will discuss all these types in detail. So direct ELISA, as its name indicates, it is in this type of ELISA there is a direct interaction among the antigen and the enzyme-linked antibody. There is nothing in between these two. So, okay. The indirect ELISA means the antigen is not directly attached or it does not directly interact with the enzyme-linked antibody, but it is attached by the primary antibody. Okay. Sandwich ELISA, as its name indicates, that antigen is actually sandwiched between the capture antibody and the primary antibody or the secondary antibody. This is the most commonly used format. And the competitive ELISA is the antibody in higher concentrations bind with the antigen, hence inhibiting the binding of the antibodies in lower concentration. So now in this picture, from you can see here that in direct ELISA, the primary antibody conjugate or the enzyme-linked antibody, which is shown in the yellow color, is directly attached with the antigen. This is the direct ELISA. In the indirect ELISA, you can see that the antigen, the enzyme-linked blue antibody is not directly attached with the antigen, but it is but there is a primary antibody in between these two. So this is an indirect type of ELISA. In sandwich ELISA, the antigen is sandwiched between a capture antibody and a primary antibody. And in the inhibition ELISA or the comparative ELISA, the antigen or the antigen in the higher the antibodies in the higher concentration binds with the antigens. So now the direct ELISA. So what happens in direct ELISA? Basically, direct ELISA is used for the detection of antigens. Okay. So in direct ELISA, there is a direct interaction of the antigen and the antibody. Serum from the blood sample is isolated and is kept in a buffer solution called PBS buffer. The ionic concentration of this buffer solution is same as that of the serum. This helps in maintaining the shape of the antigen. Then a solution of non-reducing protein such as bovine serum albumin is added in each well in order to cover any plastic surfaces in well which remain uncoated by the antigen. So the first two steps, addition of a buffer solution and an addition of a non-reacting protein, which is a BSA or the bovine serum albumin, these two steps are followed in all the types of ELISA. You have to add a buffer solution and a non-reacting protein in your wells while performing any type of ELISA. So a buffer solution basically is added to maintain to provide an environment to the antigen so that it does not distort its shape. And the non-reacting protein, which is the bovine serum albumin, is added to cover the uncoated areas of the well. So in direct ELISA, the primary antibody is which is attached with an enzyme or which is the enzyme tagged antibody is directly attached with the antigen which is coating the surface, which is coating the bottom of the well. Okay. Then on addition of a substrate, there is a chemical reaction and the enzyme is utilized and a product is formed, and then a color appears. So this is what happens in the indirect ELISA. It's quite simple that the antigen is present in the well. The antigen is taken from the sample from a human serum or the patient's sample, and then we add an antibody which is specific for that antigen. We add that antibody, the antigen and the antibody makes a complex in that well, and on addition of a substrate, you see a color. So this type of ELISA is best when we are analyzing the immune response to an antigen. So in this picture you can see here, this is the direct ELISA. This is the antigen which we took from the patient sample, or which we are detecting in a patient sample, and this is the antibody which is specific for this antigen, and this is the enzyme attached to this antibody, and then and on addition of substrate, these both will react and they will produce a color. So now the indirect ELISA. So this type of ELISA can be used to detect both antigen and antibody. So indirect ELISA can be done in two ways. Either you are detecting the antigen or you are detecting the antibody. So first of all, you all have to add the PBS and the non-reacting protein in your wells. Then you're going to add then if you're detecting an antigen, then you will have to coat the well by the antigen from the patient sample. Okay, So first, if you're detecting or you're suspecting an antigen in a patient's blood, you draw that. So you draw the serum from that patient, and you then add the serum along with that antigen or the suspected antigen into the well, and then the antigen is allowed to bind to a specific antibody for that particular antigen. Okay. So if the patient's serum contains that antigen which we are suspecting, then the antigen antibody complex will subsequently be detected by a secondary enzyme coupled antibody. So there are two antibodies involved in this type of ELISA. First is the primary antibody, which is specific for that particular antigen which is coating the well, which is present at the bottom of the well, and which we took from the patient's serum. Okay, so first antibody, which is the primary antibody, it attaches with the antigen. Then this antigen antibody complex is